Dear students, today I would like to talk about the development of the heart. I have two lectures about this topic because in the embryo exam you have a lot of questions about the development of the heart, about the different parts of this whole procedure. Here you can see the schedule of the whole lecture. First, we will talk about the establishment of the cardiogenic field. After, we will see the formation and the main parts of the heart tube, because first the heart is originated from the tube-shaped organ which is called the heart tube. After, we will talk about the looping of the cardiac loop, the different parts of it, the formation of the loops. After, of course, we have to divide this tube into atria and ventricles, and after we have to separate the common atrium and the common ventricles into two parts. So first we will see the development of the atrioventricular canal. As I told you, we will separate the atria and the ventricles, and after we will see the formation of the atria, we will see the development of the sinus venosus and the formation of the septum, interatrial septum. Of course, we will see the malformations of these steps, and after finally we will see the division of the ventricles and the formation of the septum within the truncus arteriosus to divide it into pulmonary trunk and the aorta, and finally we will see the malformations of the last two steps. Here you can see all of the main points of the two lectures about the heart development. Let's see the first point, the establishment of the cardiogenic field. Here you can see this picture about the embryo, the pre-somite stage. This is the dorsal view of the pre-somite embryo at the day 18th. Here you can see. In the middle, we will see the primitive streak. Here you can see the formation of the neural fold. What's happening in this point? Here you can see that next to this primitive streak, here we can find cardiogenic progenitor cells. These cardiogenic progenitor cells will migrate from, in the epiblast, they will move laterally and cranially, there anteriorly. Here will be the cardiogenic zone or cardiogenic plate. This zone is located in front of the buccopharyngeal membrane. I hope you know very well this picture about the cross-section of the embryo. Here you can see very well the main germ layers. The superficial part will be the ectoderm, the lower, the yellowish, this is the endoderm, and in the middle you can see the mesoderm. The mesoderm has two main parts, and here you can see the lower part is called the splanchnic layer of the mesoderm, the other is the somatic layer of the mesoderm. Here you can see that the heart development will originate from the splanchnic layer of the mesoderm. This cavity, which is located between the lateral plate parts, this is the intraembryonic cavity. And as I told you, here within this layer you can find those cells from where we'll develop the myocardial cells, and these are the cells which will form the vessels. These are the angiogenic cells. They will form smaller groups. They are called clusters. And this is what you can see here in front of the buccopharyngeal membrane in a C-shaped cardiogenic field of the developing embryo. We have to talk about the formation of the vessels first before the formation of the heart tube. Here you can see that it will start in day 19 when blood islands will be visible in the mesoderm surrounding the wall of the yolk sac. Here you can see these very special cells, the mesoderm cells, and the formation and the development of these cells, it's regulated by the FGF, the fibroblast growth factor. Because of the effect of this factor, these cells, they will differentiate and they will be the hemangioblasts. These cells, they will form the vessels. From the hemangioblasts, we'll develop two types of cells. First, that which will form the wall of the vessel. These are the angioblasts. And the hemopoietic stem cells, they are located within the lumen. From these cells will be the cells of the blood. Here you can see another factor, the vascular endothelial growth factor, which is also really important in the formation of the vessels during the vasculogenesis. Let's see now the formation of the cardiogenic cardiac question, the cardiogenic field. As I told you, this horseshoe-shaped endothelial-lined tube will be in front of the buccopharyngeal membrane. This will be the origin of the whole primitive heart tube. First, it has this C shape, here you can see, surrounded by myoblasts, and this very special part of the embryo is called the cardiogenic field. Of course, so here you can see that this structure has a horseshoe shape, and from this we have to form a Q-shaped organ, which is the primitive heart tube. Of course, for this, we need a three-dimensional movement to fuse the two lateral sides of this horseshoe-shaped structure. This will be during the folding of the embryo. Before we are talking about the folding of the embryo, I would like to tell you the two main factors which are involved in the development of the whole cardiogenic field: the NKX2.5 factor, the TBX5 factors, but also the BMP and Wnt inhibitors. They are also affected within the whole procedure. Okay, let's go and let's see the next point, the formation of the heart tube. This was the second point. Here you can see this three-dimensional picture about the position of this developing cardiogenic field. Here you can see the horseshoe-shaped structure, which is located in front of the buccopharyngeal membrane. So the cardiogenic field is located there. We have a pericardial cavity, which will originate from the intraembryonic cavity. This is the cavity which will surround this developing structure. And you can see that all of these structures, they are located in front of the buccopharyngeal membrane at the beginning of the whole development. But the embryo will grow, and we have two types of foldings during the development. The first is the craniocaudal folding of the embryo. The craniocaudal folding of the embryo, here you can see on this picture, it is because of the extensive growing of the brain vesicles, which are located here. You can see the brain vesicles, they are growing, And the developing heart, the cardiogenic field with the pericardial cavity, will move downward towards the thoracic cavity, finally thoracic cavity. Here you can see the steps. And this is the final position of it. Here you can see the buccopharyngeal membrane. In higher magnification I am showing you. The buccopharyngeal membrane is located there. Here you can see the developing brain vesicles, the developing forebrain. This is the first part of the primitive gut, the foregut. And here you can see the primitive heart tube with the pericardial cavity, which is located there of course, in the thoracic cavity. And this part, from this part, will be the thoracic cavity. And now, the first was the craniocaudal folding of the embryo. But during the development, we can talk about the lateral folding of the embryo too. Why do you have lateral folding? Because of the extensive growing of the somites on the lateral sides of the developing embryo. So here you can see the steps of the lateral folding of the embryo. On the surface, you can see the ectoderm, and the yellowish part is the endoderm. Here will be the developing foregut. On the dorsal part of the embryo, here will develop two vessels. These are the dorsal aorta, paired dorsal aorta. And there you can see the heart tube. First, it has a horseshoe shape, but during the lateral folding, these two parts, they will fuse and they will form a tube. So here you can see the steps, the dorsal aorta, what I told you. The two parts of the horseshoe-shaped cardiogenic field will be closer to each other. And finally, they will fuse. And here you can see the fusion of it. Here the primitive gut behind. This is the primitive heart tube. You can see the innermost layer will be the endocardium, and after it will be surrounded by the myocardium. And you see it very well that first this tube is fixed to the dorsal surface of the cavity. This is called the dorsal mesocardium. But during the development, here you can see the different cells. As I told you, the endocardium. Here you can see the myocardium as a special jelly. It's called a cardiac jelly. And finally, there the outermost layer is visible too. During the development, this double layer will disappear, and you can see that the heart tube is not fixed to the posterior wall of the thoracic cavity. This will be the transverse sinus of the pericardium. I hope you saw it in the dissection room in the anatomy practice. Here you can see it. It is visible around the arteries, the aorta, and the pulmonary trunk in the cadaver. So here you can see again the endocardial cells and the special myoepicardium. From this layer will develop the myocardium and the epicardium of the heart wall. The cardiac, I also mentioned the cardiac jelly. This is secreted by the myocardial cells, and it contains a lot of hyaluronic acid. This has a really important function in the development of the wall of the heart. And here you can see the position of the primitive heart tube. It's fixed. Here we have an upper pole and the lower. The cranial pole will be the pole of the arteries. Here, from here will develop the aorta and the pulmonary trunk. It will form connection with the aortic arches with the dorsal aorta. And here at the lower part, you can see the venous part of the cardiac loop, where the veins will drain into the heart, the primitive heart tube. And now I would like to show you this whole procedure in the short movie. Please check it. Here you can see the YouTube link of this video, what you can see at home. You can see it in slower manner, but now let's see it together. So the development of the heart will start at the day 18, when we can see the presomite embryo with the primitive streak and the neural fold. The horseshoe-shaped cardiogenic crest is developing. First, it is located in front of the buccopharyngeal membrane, but because of the folding, it will be behind that, the future thoracic cavity. Here, the oropharyngeal membrane, the brain vesicles are visible in the picture. But for this movement, it's really important, the lateral folding of the embryo. The dorsal aorta, they are developing. Here we can see the cardiogenic field. Because of the lateral folding, they will be closer to each other. They will fuse to form a heart tube. In the middle, we have the endocardial cells and the myocardial cells and the pericardial cells. Let's continue the next part. It will be the formation of the cardiac loop. The formation of the cardiac loop, here you can see we are in the day 22nd. Here you can see the apical pole, the cranial pole, and the caudal pole of the heart tube. During the development, we will see the looping of this cardiac loop. The upper cranial part will move to the right side downward. Here you can see the position anteriorly. Like the caudal part, from where will develop the venous part, the atria. They will go upward towards the left side posteriorly. So this is the direction of the looping of the heart tube. Here you see in the day 23rd, also it's visible that the cranial part will grow, will move to the right side downward, and the posterior part is moving upward back to the left. It will continue in the day 24. You can see this. You see the loops of the developing heart tube. In the day 26, you can see bigger loops. This cranial part, it's moving to the right side. The distal part is moving upward to the left posteriorly. And finally, here, the whole looping will be finished at the day 28th. Here you can see the first part. I would like to tell you the main parts of the primitive heart tube. If we see this position in the day 22nd, this is the form of the primitive heart tube. We have to know very well the main parts of the primitive heart tube. What are those? If we start from the venous part, there first we have to mention the sinus venosus. The sinus venosus, this is the first part of the primitive heart tube. Here will drain the main veins of the embryo, what I will tell you. The umbilical vein, the vitelline vein, and the cardiac veins, which will drain there to the sinus horns and to the heart tube. After, the next part will be the common atrium. From the common atrium will develop the auricles of the heart. I hope you remember, we saw it in the anatomy practice. I would like to go back to the sinus venosus a little. From the sinus venosus will develop the smooth parts of the right of the atrium, the smooth part of the right atrium. This was the sinus venarum cavarum. After, we will continue, and we can see there the ventricle, the primitive ventricle. From the primitive ventricle will develop the left ventricle. Because of the looping, it is going towards the left side. We will see step by step this development. And finally, the last part of the primitive heart tube is called the bulbus cordis. And we can divide the bulbus cordis into three main parts. From this part, which is number 3, from that part will develop the trabeculated part of the right ventricle. After, it's the number 2, this is called the conus cordis. The conus, from the conus cordis will develop the outflow parts of the ventricles. And finally, the truncus arteriosus. It is the uppermost part of the bulbus cordis. From this will be the truncus arteriosus. From this will develop the aorta and the pulmonary trunk. You can see very well the direction of the looping of this whole primitive heart tube. This part, the bulbus cordis, will move downward to the right side. I think that's why maybe it's logical that from this part will develop the trabeculated part of the right ventricle. This line there will demonstrate for you the position of the pericardium. You can see very well that first, within the cavity of the pericardium, we can see only just the bulbus cordis and the ventricle. But during the development, later, the common atrium and also the sinus venosus will fuse, will go posteriorly and upward, and will be within the cavity of the pericardium. Also, I would like to mention one junction which is located between the common atrium and ventricle. This is called the atrioventricular junction and atrioventricular canal. This is first the connection between the atria and the ventricles, and that is where the atrioventricular orifices will develop. Let's see now the next position. Here you can see the bigger loop of the primitive heart tube. I would like to show you again the main parts. The sinus venosus, this is the first part. After the common atrium, you can see that the common atrium is located within the pericardial cavity. You see the ventricle, the common ventricle, primitive ventricle is bigger. From this will be the left ventricle, as I told you earlier. And you see also the bulbus cordis, which is going to the right side. Here you can see the anterior view of this heart tube. You can see the position of the main parts. The sinus venosus and the atrium will be there, but they will go posteriorly. If you remember the anatomy of the heart, you know very well that the veins and the atrium, the left atrium, is located in the posterior surface of the heart, in the mediastinal surface of the heart. Here you can see that the ventricle is located in the left side, so from the primitive common ventricle will be the left ventricle, and the bulbus cordis is located there in the right side, and here we can see the two other parts, the conus cordis and the truncus arteriosus. From these parts will be the outflow part of the ventricle, and from the truncus arteriosus will develop the aorta and the pulmonary trunk. Here you can see the main steps, and it is also visible the border between the ventricles and the atria, the atrioventricular canal. This is the day 24, and the position of the different parts, you can see it very well, that the veins, they are going towards the posterior part of the whole primitive heart tube. So here you can see the sinus venosus, how the veins, they drain to the sinus horn and to the atria. After, you can see in the posterior part of this whole primitive heart tube, this is the common atrium. After, we see the common ventricle, which is located in the left side. It will form the left ventricle. And finally, it's visible there, the bulbus cordis. It has three main parts. The trabeculated part of the right ventricle will develop from this part. From the part number 2, it is the conus cordis. From this will develop the outflow parts of the right and the left ventricle. Of course, we have to divide it into right and left side finally. And there, the number 1 is the truncus arteriosus. From this will be the aorta and the pulmonary trunk. Here you can see very well again the atrioventricular canal. This is first the communication between the common atrium and the ventricle, and that is what we have to close, and that is how here we will separate from each other the atria and the ventricles. In this picture, in the anterior view of this stage, we can see another sulcus, which is called the bulboventricular sulcus. It's located between the bulbus cordis and the common primitive ventricle. Here, there, in this part, will develop the interventricular septum. So, of course, first we have there the primitive, the primary interventricular foramen, but this is exactly where we have the anterior interventricular circles, and that is how we can separate from each other the right ventricle and the left ventricle. And this is the final position, which is so close to the final shape of the heart. Let's see again the structures. You see the veins, they are located in the dorsal part, in the dorsal surface of the heart, the mediastinal surface. After, here you can see the common atrium in the dorsal part of the heart tube. After, in the left side, you can see the ventricle. From here, common ventricle, from here will be the left ventricle. And finally, this is the bulbus cordis. From the lower part of the bulbus cordis will be the trabeculated part of the right ventricle. In the middle, you can see the conus cordis for the outflow part of the ventricles. And finally, there you can see the truncus arteriosus, from where will be the aorta and the pulmonary trunk after the division. Again, the atrioventricular canal I would like to show you. Here we have to separate the common atrium from the ventricles. There will be the atrioventricular orifices. And there, anteriorly, the bulboventricular sulcus. Here will be the border between the two ventricles. Here will be the interventricular septum. The primitive left ventricle and the primitive right ventricle is very visible in the day 28th. And now let's compare this picture from the heart tube after the looping day 28 is so similar to the shape, the final shape of the heart. Which is which? I would like to compare it with you to show you the structures. So here you can see the sinus venosus. It's located in the posterior part. Here you can see the veins, the superior vena cava, the inferior vena cava, and also the pulmonary veins, which will open to the right and the left atrium. After the next part, here you can see the common atrium. From the common atrium will develop the auricles of the right and the left atrium. I did not tell you that from the sinus venarum, sinus venosus will develop the sinus venarum cavarum, the smooth part of the atrium. After you see that these parts, the auricles, they will surround the atrium, the aorta and the pulmonary trunk. So here you can see those structures in front. First, in the left side, from the common ventricle will be the left ventricle. From the bulbus cordis will be first the upper part, the aorta and the pulmonary trunk. From the middle part, from the conus cordis, the outflow part of the ventricles. And finally, from the lower part will be the trabeculated part of the right ventricle. This is really important slide for you because in the embryo exam, the examiner will ask you that from which part of the for the cardiac primitive heart tube, which part of the major heart will develop. So it is really important to know this slide and this picture. Let's see again this whole procedure in a movie. The formation of the loop. Here you can see the cranial part and the caudal part of the heart tube is fixed to the primitive thoracic cavity there. Here you can see all of the structures. During the development, the looping will start. Here you can see the cranial part will go downward anteriorly to the right. The caudal part will move posteriorly upward to the left. And here you see the day number 28. This is the end of the looping. The primitive right ventricle, left ventricle is visible. If you see everything from another view, you see this procedure step by step. The veins, the common atrium, ventricle, and the bulbus cordis. And here you can see the development until the day 28. This is the final position. You see the veins and the atria, they will go posteriorly. The atria will surround the truncus arteriosus. So there posteriorly will be the veins, the common atrium, the left ventricle, and the right ventricle. It is really important to know that the whole development of the heart will start from two different heart fields. We can talk about first primary and secondary heart fields. In this picture, you can see very well the position of these heart fields. First one is the red. The secondary heart field is located next to it. During the development, you can see in this picture, which will show the development, you can see those structures which will develop from the primary and from the secondary heart field. You see very well which parts of the heart will develop from the secondary heart field. From the secondary heart field will be the bulbus cordis, the trabeculated part of the right ventricle, the outflow part, and also the truncus arteriosus. It is really important to know that in this part, in the development of these structures, here we have to mention the cells from the neurocrest. The neurocrest cells, they are also involved in the formation of this part of the heart. That's why it is so frequent, the development of developmental malformation of the head and neck region. This developmental malformation, it can combine with the development of the heart. Let's see the next point, the next and the last point of this presentation today, which will be about the septum formation of the atrioventricular canal. First, I have to tell you how it's possible, how can we divide one whole cavity into two separated portions. Here you can see the first possibility how we can divide one cavity into two main parts. Here you can see a protrusion from the wall will develop. These are called the endocardial cushions. These endocardial cushions and ridges, they are able to grow and they can fuse, for example, in the middle, or they can grow from one side until they reach the other side. In this type of the development, we are able to separate two main parts correctly. Here you can see another type of growing, of separation. In this form, you can see one part of the wall which is not growing there, but the other parts which are located next to it, these are growing so fast, they form like an expansion. So this part which is not growing, it will remain here like a septum. But as you see, here we have not a total, not a correct closure between the two main parts. For this, we need an additional structure like a cushion, for example, which will close this communication between the two main parts of this cavity. First, the development of the atrioventricular canal and the separation of the atrioventricular canal, we will talk about the formation of the endocardial cushions, which will separate the atria from the ventricles. So let's see that. What's happening? Here you can see the picture from the heart tube. This is the atrium located posteriorly. Here is the left ventricle and the bulbus cordis. And there you can see the atrioventricular canal. In the atrioventricular canal, you see that endocardial cushions, they start to develop. Here you see there how it's happening. You can see the myocardial cells and the endocardial cells which will form the innermost surface of the heart tube. These myocardial cells, they will produce some inductive factors which will stimulate the endocardial cells, and they will produce other mesenchymal cells, here you can see, which will proliferate and will grow and will form a protrusion on the wall. So this is the procedure how the endocardial cushions, they are developing there in the atrioventricular canal, but we can see the similar structures in the outflow part and the truncus arteriosus of the heart tube, because we have to separate here also the outflow parts and the aorta and the pulmonary trunk from each other. If you see this picture, it is visible that first this atrioventricular orifice, it's located in the left side of the whole tube between the common atrium and the common ventricle, from which will develop the left ventricle. But during the development and the formation of this inner curvature, which is called the bulboventricular flange, here you can see that this opening will move from the left side to the right side, and finally it will be in the middle, so will form communication with the common atrium, with the ventricle, and also with the lower part of the bulbus cordis, from where will develop the trabeculated part of the right ventricle. And here we see the cushions. We can talk about superior endocardial cushion, inferior, and we have two lateral ones. They will grow and they will fuse. Here you can see the fusion of the superior and the inferior cushion, and this fusion will separate the atrioventricular canal into two parts, to the right side and the left side. So here you can see the picture. This is how these cushions, they will fuse and they will form a separation between the right side and the left side of the atrioventricular orifice. In the right side, there in the right atrioventricular orifice, here will be the tricuspid valve, and in the left side, there will develop the bicuspid valve. You can see the blood flow. From the right side, we are going to the pulmonary trunk. From the left side, we are going to the aorta. And finally, here we can see the development of the valves, the tricuspid and bicuspid valves. First, we have a connection between the cushions and the muscular wall of the heart. And because the blood hollows these parts of the muscular wall, these muscle fibers, they will disappear, and we have only just there the valves, which is just connective tissue, you know, the endocardium and the little connective tissue. Here you can see the chordae tendineae, and at the lower part we can see the muscle, which is the papillary muscle, which will fix the chordae tendineae to the wall of the ventricle. And of course there you can see also the trabeculae carni. Let's say this, finally the whole procedure. The primary and the secondary heart field, they are visible in this movie, and you can see very well that from the secondary heart field will develop the main part, the bulbus cordis, the outflow region of the heart tube. There you see very well the bulbus cordis, trabeculated part of the right ventricle, the outflow part, and the truncus arteriosus will develop from this. If we see the division of the atrioventricular canal, there we can see the developing endocardial cushions. They fuse, they grow, so the myocardial cells, they produce these special inductive factors. The endocardial cells, they produce new and new cells, which will grow and will form a protrusion on the wall, and will close, will separate from each other the atria, the common atrium, and the ventricle. This is the fusion in the middle. The same structure will be also in the outflow part. First, this atrioventricular orifice is located in the left side, but during the development of this bulboventricular flange. Here you can see we have a changing within this part, within the bulboventricular region. That's why this whole atrioventricular orifice will move from the left side to the right side, and finally will be in the middle, will form connection between the common atrium and the ventricle and the trabeculated part of the right one. From here we have to the pulmonary trunk, from there to the aorta. And this is the way how the endocardial cushions, they will meet and will separate from each other the right side and the left side. In the right atrioventricular orifice, here in the right side, will be the tricuspid valve. In the left side will be the bicuspid valve. Here you can see the formation of the cuspid valves. First we have muscle fibers within these connections, but after, from this will develop the leaflets, the chordae tendineae, and the papillary muscle. So this is what I wanted to tell you in the first lecture. I will continue this whole procedure in the second presentation. Thank you for your attention.